Hi, this is Miss Litton, and this is a Friday afternoon with a room full of very dedicated students that I'm very, very proud of. Say hi. Hi. Oliver. Oh, wait, we got two. Two men in the room. Excellent. Really proud. Yay. Good job. All right. Now, I'm going to re-remind you, we have already reviewed this Unit 2 before, and that is all already recorded the first time we did it, right? So I'm going to kind of hit them kind of fast and hard, but here is a really good list of things that we need to remember from Unit 2. So let's see if we can do one, and then we'll look at some pictures about it. Earth structure. What are some things we need to remember about the Earth structure? Hey, we got another man. Yay! Where are all your homies?
I don't know.
So disappointing. Okay. Okay, Karen. All right, so we have, what do we have at the core? The core. Yeah, we have the core. We have the core at the core. What's outside of the core?
Outer core.
Okay, good. What's outside of that? The mantle. Here, let me get you a picture. Yes, here we go. All right, so we've got our solid core, our outer core, then we have our mantle. Yay!
So how many?
What do we have? 4 now.
Yeah.
Okay, I don't know if it's statistically significant, but I think it is. Okay, focus. Yay! What do we have going on in the mantle? Convection currents. And then those convection currents are moving the plates. Good. All right. So the way that guys, don't talk when you come in late. How that could play itself out is we could have the crust going away from each other. What do we call that? Divergent. They're going away. Yeah, divergent boundary. And we could see that like in the ocean, it could happen, and then new ocean crust is formed. But could that also happen on land? Yeah. Remember I showed you that video and he was swimming through it? Do you remember that? Maybe. Okay. And then what happens if an ocean crust meets a continental crust and they're headed for each other? What do you think is going to happen? Ocean crust is going to go under and the land is going to be on top. Yes? And that's why the ocean crust is younger material than the land, right? Right? Because the ocean keeps getting reformed like this and the land just stays on top. Yeah? What happens if two continental crusts come toward each other? What's going to happen? Mountain. Okay, good. So we looked at divergent, we looked at convergent, and then one we have here in California is the Pacific and the North American plate headed in two different directions, which then causes what? Earthquakes. Good. All right. We looked at all the different evidence for this. When you look at the seafloor spreading, that we know that the north and south poles have reversed. Because the way the minerals are forming within the seafloor, we can tell which way is north, which way is south. Yes? Yes?
Wait, what was that last one called?
Transform. Here. Got it?
Yeah.
Okay. Also, we can see that there's newer crust material right here at the seafloor spreading as far as the age of it, right? We can tell that. I don't know. Oh, yeah, here we go. We looked at Pangea. And now, based on what we're learning right now in the chapter 27, look right here with the fossils. Do you see how that's, remember what we were talking about on biogeographical evidence for evolution? That if the continents are together, then you expect to see these species be able to migrate around. But that when the continents had all come apart, that was about 65 million years ago, and the species that were evolving 65 million years ago were what? Come on, mammals, thank you. So that's why we see such species diversity or species-specific mammals on the different continents. Got that? Okay. You're still right, your brain's in gear because you're maximizing today. It does you no good if you're just doing seat time. I want your brain in gear so we can use this time well. All right. Then we talked about how as the earth changed, then that could affect what organisms could live there. And then those organisms living there could impact what happens to the earth in that place, right? As you start to have grasses grow and then bigger plants, then when they die, it creates more soil and you could have other plants that are growing there, yeah? We talked about how the evolution of non-cyclic photophosphorylation and the oxygen, revolution created a what? Ozone layer. This allowed then life. You could go from unicellular to multicellular. Right. So you have this cycle, okay? And I think, yeah, let's talk about early Earth. We've already talked about this. We just reviewed this in the last one, right? When we talked about the early Earth, the gases that were present probably came out of a volcano. What gas is missing that we have today? Oxygen. Today we have an oxidizing environment, but in the early Earth we had a reducing environment which was conducive to building. Excellent. Okay, so here are those early gases. Yay. Okay, and then we looked at the steps of life. And we just got through reviewing this. Inorganic molecules coming out of the volcanoes. What energy do we have to build these organic molecules? We have UV radiation. We have lightning. We have magma. And we have radiation. And then you could go from these inorganic molecules to organic. And there was a man who did an experiment. Say my name, say my name. Miller. Okay. Then you just need these things to hook together to form your polymers, right? Monomers to go to polymers. And so in order for that to happen, same energy as we talked about before, but also the surfaces of some rocks could act as a catalyst. What are those rocks' names? Clay. Good, you got it. Then the first cell we have, prokaryotic or eukaryotic? Prokaryotic. Autotroph or heterotroph? Heterotroph. Unicellular, multicellular? Unicellular, right? And then how could we have had a eukaryotic cell evolve from a prokaryotic cell? Name the hypothesis. You know it.
Endosymbiotic hypothesis.
Yes, endosymbiotic hypothesis, which says one prokaryote went to live in another prokaryote. Evidence for that? The energy organelles. Who are the two energy organelles? Mitochondria and chloroplast. They have their own DNA. It's in the shape of a circle, just like prokaryotic cells. They are they have their own ribosomes, and they're like prokaryotic ribosomes. Also, they have how many membranes? Two. And then what do we call this when the membrane? Invagination. It could infold, right? And move around and enclose the nucleoid region of the prokaryotic cells in order to form a nucleus. Does that all sound familiar? Vaguely. Vaguely I know about that. All right. Then we go to chapter 1. We totally know this one. Let's do this real quick. Here we go. Living things are organized, made of cells. They acquire materials and energy. They maintain homeostasis. They respond to stimuli and reproduce. They grow and develop, and they adapt and die. Good. When we said living things are organized, made of cells, how are they organized? Atoms, molecules. Stop. Molecules. What are the three important inorganic molecules? Water, oxygen, carbon dioxide. What are the four important organic molecules? Carbohydrates, lipids, proteins, nucleic acids. Atoms, molecules. How do we go from atoms to molecules? They have to form bonds. What were the two big bonds we talked about? Ionic and covalent. Which one is about ditching and stealing electrons? Ionic. Which one is about sharing electrons? Covalent. And when you share in pairs. If I share one pair of electrons, it's a single bond. If I share a pair, two, it's a double bond. If I share unfair, what is it called? Polar covalent. Polar covalent. And is that going to be hydrophilic or hydrophobic? Hydrophilic. Because if it's polar, it could have a partial charge on it, and water can deal with that, right? What is it when they share fair? Hydrophobic. And if you're hydrophobic and you're sharing fair, you're sharing fair and you're hydrophobic, that's because there is no charge on you, and water, who is a polar molecule, can't connect. Okay, tell your bio buddy, was there any part of that you're a little sketchy? Right? Okay, so we did that. We talked that all life could be put in three baskets. Bacteria, archaea, and eukarya, right? All life can be put in those. Bacteria and archaea have in common that they what?
They are unicellular.
They're both what?
The thing that came first.
Archaea.
Bacteria and archaea are both prokaryotic, right? What's different, but you did it. But what's different about archaea? They are streamophiles. Good. And are archaea newer or older than bacteria?
Newer.
Newer. Yeah, they messed up the name, right? They called them archaea, which means ancient bacteria, but they're not. They're closer in many things to eukaryotes than they are to prokaryotes. All right, good. And then when we look at the eukaryotes, we will simplify them just into 4 kingdoms. And those are the posters on the right? So Protista is generally unicellular, but they're eukaryotic cells. Okay, they can be autotrophs, they can be heterotrophs. Fungi are absorptive heterotrophs, where we as Animalia are what? Ingestive heterotrophs. And then Plantae are what? Not heterotrophs, but autotrophs. Remember reading that? Okay. Then we went into the scientific method. Okay. So we do that all the time in all of our labs. So the first thing we want to do is what? Make some observations. We want to ask a question. Then we'll do a little bit of research. We'll look in some books. And then, right, that's why we do a background, right? Driving question, background. And then we write our hypothesis. Okay. Our independent variable is what we change, right? Our dependent variable is our dependent variable is our data. Dependent variable is your data, right? If I give my plants more light, then they will grow tall. Okay? So what's the independent variable there? Amount of light. What's the dependent variable? How tall they are, their height. What could be my control? No. Those are constants you just gave me. What's my control? No light or 100% light. Either one of those could work. What would be constants? I would use all the same plant, all the same soil, all the same amount of water. Those are constants. Constant in everything. The only thing I change is the independent variable, which is the amount of light. Got that? Constants and control. Your control is your basis of comparison. Okay? Control is your basis of comparison. All right, when I do my, what am I going to do after I do my hypothesis? I'm going to do my experiment, okay? And in my experiment, I could have 2 kinds of data. What's my 2 kinds of data? Quantitative and qualitative. Which one can I graph? Quantitative, good. Then I'm going to do my analysis, right? Claim, I can evidence, reason. I'm going to make a claim based on my evidence, and here's the reason why. Plants grow better in more light. My evidence is here's the height of my plants. The reason is they need the light to do the light-dependent reaction, so they can do the Calvin cycle, dark reaction, light-independent reaction to make more sugar so they can grow taller. See how I can work that out? Okay, and then I make my conclusion based on answering my driving question. All right, so we talked about independent, dependent variable already. The atom story. Atoms are made of protons, neutrons, and electrons. The protons and the neutrons are in the nucleus, atomic mass. The electrons are in energy levels. 2, 8, 1832, 8, 8, 8 is great octet. We know one atom hooks up with another atom in order to get happy. How can we make them happy? Either by filling it up or getting it to 8. Do they mind ditching and stealing? No, they do not. Okay, do they mind sharing? No, they do not. Okay, what do we call it when they share? Covalent bonds. What do we call it when they share fair? Non-polar covalent. When they share unfair, it is polar covalent. I asked you that twice because I want to make sure you get it. All right, is lithium happy? No. Okay, how many protons does lithium have?
7.
Really?
Oh, wait.
3. Thank you. How many neutrons does lithium? See how well I teach it? How many? 4. Neutrons equal 4 because the protons plus the neutrons equal the what? Atomic mass. Okay, how many electrons do I have?
3.
Good. Okay, because on your periodic table, it's going to show you balance. That's all you need to know for this point. Now, I need to find those electrons home. How many can fit in the first level?
2.
Oh my gosh! How many can fit in the next level? 8. How many can fit in the next one? How many in the next one? Thank you. How many electrons do I have? 3. How many are going to play in the first level? 2. How many in the next level? 1. There we go. So this tells me how lithium is going to behave. He needs to share with somebody who has 7, or he could ditch it, right? So these are the things, this tells you what it's going to do. And in fact, if I look at a periodic table, I'm going to find lithium, and he's going to be right here. Do you remember we had 2 things, the columns and the rows? What are the rows? Periods. You know how you remember that. You're on your period, okay? So you have to lay down, okay? These right here, oh, it's listed. You knew, it's on my graphic. You're like, that's periods and groups, I know. Okay, so this entire group all has the same number of valence electrons, and what is that? 1. Okay? So they will all behave in the same way. You can predict their behavior and their reactivity with other molecules based on their number of valence electrons. When we look at this right here, this is a period, they are all at the same what? Energy level. Yeah. Okay. You're going to learn more about that in chemistry. Now, isotopes are when you vary in the number of what? Neutrons. Ions are when you vary, isotopes the number of neutrons, ions are electrons. Good. We already went over this. Sharing. Okay. Sharing is caring. Hydrogen bonds. When one water's oxygen, which is partially negative, is attracted to another molecule's hydrogen, which is partially positive, then that is a weak but important bond called a hydrogen bond. Where do we see that in other things? It doesn't have to be with water. It could be 2 chains of nucleotide, 2 chains held together in DNA. Okay. Next, we went over the properties of water, and those properties of water all have to do with those hydrogen bonds, right? High heat capacity, high heat of vaporization, ice being less dense than liquid water because as it cools, it forms all the same equal distance in their hydrogen bonds, right? Remember all of this? Being a good solvent as long as it's a hydrophilic substance. Those are all properties of water. And we also referred to the pH, right? pH of a substance is a measure of what? Hydrogen ions, yeah. And so if you have a low pH, though, we say that you are a acid. And if you have a high pH, you are a base. Good. Okay? Then we went into the 4 important organic molecules. Now, the first thing is when you hook 2 amino acids together, or when you hook 2 sugars together, what's the name of that reaction? Yes. Boom shakalaka. Dehydration synthesis. What's another name for it? No. Condensation synthesis. It's okay if you don't remember that. Dehydration or condensation synthesis. Because what's happening when you build is, look right here. This sugar molecule is totally happy by himself. This sugar molecule is totally happy by himself. If they're both happy, why would they ever hook up, right? So we need to take something away from both of them so they can get happy together. Okay? So we take an H from one and a OH from another, and that forms a water molecule. Dehydration synthesis, because we're dehydrating, we're removing a water. Water is going to be on the product side. Condensation synthesis, when something condenses, like if you have a glass and it's cool and the air hits it and you get the drops of water on it, your mom says, put a, thank you. Condensation synthesis, you're still forming water. Water is on the product end of that reaction. If water is on the reactant side, you're probably not doing dehydration synthesis, you're probably doing a hydrolysis. And we know to facilitate any of these reactions from going forward, you probably need an enzyme to facilitate that process. What do enzymes do? I'm jumping ahead, but enzymes lower the energy of activation to make the reactions more likely to go forward. That's not in this unit, but you know it now, so we can address it, right? Okay, so then it's just a matter of knowing your building blocks, like sugars always build carbohydrates. And remember, a simplified sugar, we can simplify them always down to CH2O, carbohydrate, right? So if I have a 6-carbon sugar, it's C6, then 2 times that is 12, and then O6. Does that make right? Remember that? Okay. So then we talked about that when you build polysaccharides, you could have disaccharides. I just skipped over that, but you remember the disaccharides? Sucrose is table sugar. It's glucose plus fructose. Did I have you learn those? Yeah. Maltose is glucose plus glucose. Lactose is glucose plus galactose. Lactose is like milk sugar. Okay. Fructose is fruit sugar. Sucrose is table sugar. Polysaccharides is when you put a whole bunch of sugars together, and there's 2 functions for that. It could be energy, like starch or glycogen, right? We do glycogen, plants do starch. Or it could be structural, something you're not really eating, but it's a good way to build a wall, like a cell wall, like cellulose. Or chitin, which is fungal cell walls. Okay, so it could be either or. Lipids. Lipids don't have a single brick, and in fact, the lipids don't have a single category, because lipids, under the lipid umbrella, what are our 2 options there? Fats and steroids. Okay, let's focus on the fats. The fats are made of 2 things. Glycerol and long fatty acid chains. The fatty acid chains are hydrocarbons. Say it? Hydrocarbons. And those hydrocarbon chains, if they're in animals, they're probably a what? Saturated fats, all single bonds, no double bonds, right? All single bonds. They have straight chains, and that's why they stack up as solid in room temperature. Unsaturated fats are found in plants. That's why you have plant oils, exception palm oil. And so as a result, they are liquid at room temperature because their fatty acids have double bonds in them, and it puts kinks in their chains. And they tend to be a liquid at room temperature. Okay, if you can have, here's that too. This kind of explains it. Then, are you glad you came today?
Yeah.
Are we reviewing a lot? Yes, okay. Be brilliant. Now, you can have a modification of one of these fats, and that is if you remove, right, if you remove one of these chains right here. If you get rid of that one chain and you replace it with a phosphate group, then you will have a phospholipid, and phospholipids make up cell membranes. All right. Okay? The other thing, if you're not a fat, you are a steroid. And steroids are characteristically made out of 4 rings of carbon. Yeah, 4 rings of carbon. Okay? Proteins. The building block for proteins are amino acids. Amino acids always have the same structure. A central carbon, hydrogen, a carboxyl group, an amino group, and below they have an R group. An R is not any one amino acid. An R group is one of how many different options? 20, so therefore you have 20 different amino acids. Those R groups could be polar, non-polar, right? Hydrophilic, hydrophobic, charged, not charged. And so that affects, when you put a big chain together, how they fold. And the shape that a protein is in affects its function. The most dramatic one I could tell you is all your enzymes, right? Your enzymes have to be in a correct shape in order for them to have their what? What's the key part on an enzyme?
Substrate.
Where the substrate binds. Active site. The active site has to be in the exact right shape. If it's not, then the substrate can't bind to it and the enzyme can't do its job. So what shows, you have these chaperone proteins that fold it, but if you have a hydrophilic amino acid where you should have a hydrophobic, it won't fold correctly. Okay? Next we talked about nucleic acids. Nucleic acids are built out of nucleotides. Nucleotides consist of a base, sugar, and a phosphate group. If your sugar is deoxyribose, oh darn it, yes. If your sugar is deoxyribose, then you know that you are in DNA. If your sugar is ribose, you know you are RNA. What could be 2 other ways to know whether you're in DNA or RNA?
Whether you have thymine or uracil.
Whether you have thymine or uracil. Excellent. Because uracil is found in RNA and thymine is in DNA. Okay? D-thies. Okay. What's another way? One more. RNA is single-stranded and DNA is double-stranded. DNA double. Okay. DNA is double-stranded. Okay. Here is a picture of DNA. And then ATP. Oh, here's some RNA for you. And then ATP right here. Adenosine triphosphate. That's also a form of a nucleic acid. And then we have this chart right here. The summary chart that we have. Okay. Then atoms, molecules, organelles, cells, right? So then this brought us to, have you got it? Tell me when you have it, and then I'm going to move it. Yeah? Okay. So then we had the cell theory, and that is all living things are composed of cells. This is a big old unit, huh? Cells are the structural and functional unit, and cells come only from pre-existing cells. So we know that cells are small because they need a large what? Surface area per volume. Yeah, that's why cells are small. We looked at prokaryotic cells, and we looked at things that they don't have a nucleus because they don't have any membrane enclosed organelles. They don't have a nucleus, they just have a what? Nucleoid region. Nucleoid region. And that's just that area where their DNA is. They do have workbenches for protein synthesis, and what's that called? Ribosomes. They do have that. They have cell membranes because all cells have cell membranes. But outside of their cell membranes, they also have a cell wall, and outside of their cell wall, they have a capsule or a glycocalyx. Okay? And on here, there's part of their outside, right here, helps them attach. What do you call those? No. Fimbriae. What does the flagella do? Helps them move. And then what's this sex pillows for? For sex. Okay? So that's how they exchange DNA from one bacterium to another. Prokaryotic cells are smaller than eukaryotic cells, so that means they have a larger surface area per volume. Good. All right. Eukaryotic cells. You have plenty of pictures, okay, that you should know, okay, where you identify the structure and you identify the function. Chloroplast does photosynthesis. Lysosome is the stomach of the cell, right? Remember all of those? Ribosomes, protein synthesis. Okay, so I would recommend getting a chart or some sort and then just reviewing those organelles. Okay, and then let's focus on the cell membrane. It is a phospholipid bilayer, yes? Okay, that phospholipid bilayer has phospholipids in it, but what else is in the cell membrane? Proteins. Do you remember that? Let me see if I have a picture. Here you go. Sorry. Let me help you. Okay, so you have, this is really such a hunkin' unit. Okay, so you have phospholipid, you have proteins. What are these things right here? These purpley things? Cholesterol, those are steroids, yes. Okay, and they help with the fluidity of the membrane. If you remember, it keeps it when it's really, really cold outside, it prevents it from freezing. It was really hot, it prevents it from not melting, but like breaking apart, yeah. But you could say melting. Okay, what are these yellowy things out here? Yeah, glycocalyx. It's either sugar chains attached to a lipid, and if so, what would we call it? A glycolipid. Or it's sugar chains attached to a protein, and we call it a glycoprotein. Good job. All right. Then we talked about, how do we cross this membrane? We know how to cross it. We can go through the phospholipid bilayer, but not if you're large or charged. You can use a channel or a carrier or whole membrane endo. Well, what was the other one I really liked? Yes, receptor-mediated endocytosis. Good job. Then we said there's two kinds of transport. We talked about active versus passive. Before I go to that, I digress because I didn't talk about all the protein functions. What are all the different protein functions? They could be channels, carriers, cell recognition, receptors, enzymatic, and junctions. Are you remembering all of these songs?
Yes.
Okay. All right. So, passive versus active transport. Passive takes no energy. Active takes energy, right? And so, when it's passive, you're just going from a higher concentration to a lower concentration. Okay? If it is active, you're going from lower concentration to a higher concentration. You're going uphill because you're cramming them to one side of a membrane. Okay, and that is going to take some energy. On the passive, if you just go through the phospholipid bilayer, that's straight up diffusion. But if you use a channel or a carrier, and you're going with the concentration gradient using a channel or a carrier, then we call it what? Facilitated diffusion. Facilitated diffusion. Okay, good. Then we start talking about osmosis. Osmosis has three components. I'm giving you a hint. No? Osmosis. What do we call that word when you go from higher to lower? Diffusion of water across a semi-permeable membrane. That is osmosis. Diffusion of water across a semi-permeable membrane. And we remember, water must flow from the hypo. I know that one. Okay. So it's going from a hypotonic environment into a hypertonic. What makes it hypertonic? It has more osmotically active substances. So water's just going from a higher concentration to a lower concentration. And that's what's happening here. Okay? And it's going to raise, what's going to stop it from going any higher? Gravity. So smart. So then we look at what can happen. Here's an isotonic, same. Here's a red blood cell, plant cell. It's a biconcave disc. Looks great, looks healthy because the same amount of water going in is going out. But if you put that same red blood cell in a hypotonic solution, so it's hypotonic outside of it, water must flow from the hypo and it'll go in and swell and burst. Hypertonic, water must flow from the hypo. All of a sudden, if the solution around the red blood cell is hypertonic, that means that the red blood cell is hypotonic. Water must flow out of it and it'll shrink. Okay? We talked about endocytosis and exocytosis and receptor-mediated endocytosis, and oh my gosh, you did it. I am so proud of you. Look at me. So proud. Because today is Friday of a long week. This is one of the hardest units, but you did it here together, which, don't you think this would have been better with us than by yourself? Okay? So I'm proud of you. Feel proud. Feel proud. Do you have any questions you want to ask me about this? Yes. I can't hear her. Shh, go. Yes, that step. You want me to find that?
Yeah. Can you just talk about the first half of it?
This one right here?
Yeah.
Okay. So these molecules right here probably came out of a what? Help her out. Came out of a what? Volcano. Okay. What gas was missing there that we have today? Oxygen. So how could these gases have, oh my gosh, guys, can you help me? I told the janitor I will put the chairs up. Can everybody do that?
Wait, how do we do that?
Yes. I'm going to explain it to you, but I'm going to have everybody put up the chairs real quick, okay? Sorry, I hope you're studying. I'm going to go over this in just a minute. Okay, so how do we go from these inorganic molecules to our, because we got to have organic molecules if we're going to build a cell, we need energy. Where do we get the energy from? UV radiation, lightning, magma, yeah, radiation. Is that what you need help on? Okay, what else? Yes?
Exactly is a valence electron.
The number of electrons, valence electrons in the outermost energy level. Okay, what else? Yes?
What are the three most important inorganic molecules?
Three most important inorganic molecules?
Oxygen,
Isn't that not
carbon dioxide,
Inorganic, yeah.
water.
But that was before the oxygen.
Okay, no, if you're asking me now what the three important inorganic molecules are, it still doesn't matter. It's going to be oxygen, it's going to be carbon dioxide and water. That's important to life now, because we have life now, and we have an oxidizing environment. What else?
Exo and endocytosis are still active, right?
Still active. Good
I don't know
point,
why
because you have to
water
use
is
the
active.
whole membrane. That's going to you're moving your cytoskeleton around to do that. So that's going to take a whole lot of energy, endo and exo.
I don't know
What
why I
else?
thought it would change from one year to
The 4 important organic ones.
4 important organic, carbohydrates, proteins, lipids, nucleic acids. The way you're going to remember that is you eat 3 of those. Carbs, proteins, and fats, and then what's inside of a nucleus? Nucleic acids. You eat 3 things, and then really important in your nucleus. Those are your 4 important organic. What else? Okay, I love you. I'm proud of you. Have good weekends. No sex, no drugs, no alcohol. What happens in Vegas? No. Comes home when your penis is retarded. Have I taught them nothing?
They're not new.
All right, make good choices. I love you.